So this is about personalized diagnosis and treatment in autism spectrum disorders. Essentially, I'm just going to bring 3 cases that I think are kind of interesting in the clinic. So, yeah, and my supervisor is Dr. Stephen Jones, great person, and we're at the Genome Sciences Center at BC Cancer. And I respectfully acknowledge that my place of work is on the Musqueam, Squamish, and Tsleil-Waututh Nations. So autism spectrum disorders, it's a very variable phenotype, but it always consists of deficits in social communication and repetitive patterns of behavior. And there's oftentimes comorbidities such as intellectual disabilities and GI. So right now in Canada, 1 in 50 children have been diagnosed with autism, and there's a heavy sex bias, with 4 out of 5 of those being male. So we know that there's up to 90% of genetic contribution, but we know that we don't know a lot of what that genetic contribution is, because in only 2 to 4% of the cases is there an obvious rare causal aberration. So there's hundreds of genes that have been associated, thousands of variants, but we are unsure what causes it, and reducing the age at which children can be diagnosed is known to improve ASD scores and quality of life. Long-read sequencing, what I'm trying to take advantage of for this project is the use in repetitive regions that might previously have been unmapped, phasing of alleles in big chunks to haplotype 1 and 2, the complexity of structural variants, and the use of DNA methylation. So these are the clinicians I work with, Dr. Suzanne Lewis and Dr. Anna Maria Richardson. And this is individualized treatment for autism response using genetic environment targets, so the iTarget project, where we have 500 trio families that have been previously sequenced, but these are refractory cases with no known cause. And so we pick some of the most severe children. And just for the analysis component, just base call, half the cohort's Guppy 5, half is Guppy 6. Sorry, Dorado. Align with Minimap 2, and then just do SNP and indel calling, structural variant calling, copy number, tandem repeats, methylation, anything I can do to try and get some sort of cause. And then we use IGV to make sure it's real, and the prediction part is, I think, the hardest part, where we're looking through these databases. Nomads add more structural variants recently, but it's still pretty difficult. So yeah, structural variants, Sniffles 2, I've always wanted to meet Fritz, so if he's here someday. Case 1 is a young female with severe ASD, mild to moderate ID, behavioral and sleep dysregulation, anxiety, tics, language delay, and prior regression. So there was short read called a 70 kilobase pair maternal copy number gain, including ARHGF9. And this was really caught my eye because ARHGF9 has been previously been shown in cohorts to associate with intellectual disability and autism. And so when I used long reads, we could see that actually this wasn't a simple duplication. It was actually quite a complex variant. And so what I'm describing here is actually, this is on the negative sense strand, ARHGF9 has a partial duplication on the 3' end of exons 9 to 12. Cool. But also has this really crazy insertion that's been inverted from 20 megabase pairs downstream, presumably because of these homologous breakpoints. So what we've got is actually this D region inverted in between the extra copy of this B region. And that's something we never would have been able to find where this copy number increase was, first off. If it's associated with B or D, we wouldn't know if it's at these first breakpoints or at these later breakpoints, and we would not be able to tell that these variants were on the same allele. So this was a big thing to me, was the phasing, because we can take these SNPs and we can say, okay, all these SNPs are in this region, and they are associated with allele 1. So if there's a structural variant that's also associated with this allele, it's certainly on allele 1 versus allele 2. So we've got these haplotype phasing blocks. And if you see that your structural variant is within the block, both parts of it, then you know that this complexity is all on the same allele. And this is what I'm trying to show here. So these are breakpoints 1 and 2. And if you look at the break part, you can actually see that they're colored dark on either side. The break is dark. And since these reads have been associated with the 3rd and 4th breakpoint, we now know that this really complex thing is all on the same allele. And that's just a really cool thing. So then we were like, okay, how do we think that this ARHGF9 aberration functions? Well, females are mosaic. That's cool. And they experience random X chromosome inactivation at the 6 to 16 cell phase. So sometimes there is an aberration which causes skewing so that there's more non-random X chromosome inactivation, and you see one X prevalent more than the other. So all I did here is, these are actually 30 controls, but I looked at the CpG site methylations across the X chromosome promoters to see if they were like 50% allele 1 and 50% allele 2. And actually our sample showed pretty normal distribution. And ARHGF9 also had normal random X chromosome inactivation, sorry. So this is actually not the cause of disorder in this person. So this is case 2, a young male with ASD unspecified, no ID or seizure, and self-injurious behavior. He actually has it quite rough. And we were trying to figure out if maybe there was this duplication that was associated with SAE1 and ZC3H4. So actually what we found was there's a 7,000 base pair, like 5 times copy number gain in between these really critical ASD-associated genes. And so this is a really big expansion we wouldn't have been able to see in short reads. And on the same allele, we get a full duplication of SAE1. So both of these genes are not present, loss of function aren't present in the population. I assume you do not make it to birth. And they have really high Safari scores. So I was curious, do we think that this SAE1 has like 2 functioning full copies? And so I just looked at the unmethylated promoter regions, and I did see under the SAE1, the blue, it's across all reads. So there are 2 functioning copies of SAE1, which is just interesting. And so I just looked at the Cancer Genome Atlas, and I was like, do we think that increased SAE1 copy number really means increased mRNA or protein function? And really all I can say is that there's increased mRNA associated with increased copy number. SAE1 simulates proteins, so I don't know if the targets would experience increased simulation, but this is very prevalent in the brain, this transcripts, and it could be like contributing to disorder. And then this is just case 3. So this is a male with simple ASD with intellectual disability and behavioral problems. And again, we thought this was a paternal duplication. And just seeing the complexity that we can really derive out of these variants is really cool. Like, one thing that we were able to experience with phasing is this. There is a breakpoint right in the middle of anchored 11. So does that mean that this individual has 1 functioning copy of anchored 11 or 2 functioning copies using short reads? Because we can't tell if a copy going through is on the same as a copy going part way through anchored 11. Well, what we found is actually that this person does have 2 full functioning copies of anchored 11, and alongside that a 3' exon 7 to 16 duplication with these extra parts, not only this ABC that's present downstream, but an extra component of this B region as well. So very complex, and helps us figure out, just the importance of these structural variants. The last thing that I'm trying to work on is short tandem repeats. 2 to 6 base pair repeats. I use Straggler, which is present in the CARD pipeline, and they work with us at BC Cancer. And essentially, if there's a really large amount of these repeats, they can be disease if it's upstream of a promoter. So how can I find if these are in the right genes? Well, all I do is pick the longest repeats I can find in the genes that's associated with ASD, and I interrogate them in databases to see if they might have value or contribute to disease. And it's still an ongoing thing, but yeah, we'll see if I can find anything. So thanks so much, and we can find precision diagnosis. Thanks for having us. Thank you to the patients, and thank you to our sponsors, and yeah, especially Dr. Stephen Jones for letting me be here in Nanopore. Thanks.